So then we have this concept of membrane solubility. So if we have our lipid bilayer, right, when we talk about membrane solubility, we're talking about how easy is it for a thing to go from the outside to the inside or the inside to the outside, right? So for example, in the absence of any channels or other proteins, sodium is very impermeable to the membrane. In other words, if you put a lipid bilayer in a beaker and add a bunch of salt, the salt is not going to go through the membrane, all right? Because it has a positive charge. That charge is repelled by the hydrophobic region in between the cells, okay? We need, in our metabolism, to move things across the cell membrane. So we have to have ways to affect membrane solubility, and we do. For our polar molecules, there are a variety of special proteins that increase membrane solubility. In other words, make it so things can cross through the membrane, right? We have channels. Channels are passive. They're a little bit like doors. When the gate is open, molecules can move down their concentration gradient. So for example, we're going to talk a lot about sodium channels this week. And there are a variety of different kinds of sodium channels, but when a sodium channel is open, because of the concentration gradient, sodium flows into cells, right? So that's an example of a channel. Now, what causes these channels to open or close can be a wide variety of things. It depends on the protein. So some proteins open in response to voltage differences. Some proteins open in response to a ligand being present. Remember biochem, a ligand is a molecule that attaches to a protein and makes something happen, right? So a ligand fits into a receptor, just to remind you about the vocabulary. Some channels are actually triggered by physical change. You know, we appreciate these. Our sense of touch uses physical changes to the membrane to open and close ion channels, which allow us to detect our environment, right? There's lots of other examples of these, and we're going to see oodles of them as we go through this semester together. But those are channels, okay? Channels allow for diffusion, movement down the concentration gradient, right? So never backwards, okay? If you're going to move something from low concentration to high, you need a pump for that. If you want to go from high to low, you just need a channel or a facilitated diffusion molecule. Okay, so what's that? What's a facilitated diffusion molecule? It's like a complicated channel, right? So a channel allows very much free flow of a molecule when it's open. A facilitated diffusion molecule has to actually change its shape in order to allow the substance through. So it's a little bit like an airlock, right? So an airlock is where you have two doors, and you can open one door, make the pressure the same, and then open another door. So it's like a two-step process. Facilitated diffusion works like that. But because it has to change its conformation, facilitated diffusion usually is a rate-limiting step. In other words, it's saturable, to connect to a word that we talked about late last week, where adding more reactants doesn't necessarily speed up the movement because all the molecules are moving as fast as they can. The glucose transporter in the kidney that we talked about as saturable, it's this system. It's a facilitated diffusion system. Okay, then the third type of molecule that moves things through the membrane is a pump, also called active transport. In these molecules, an ATP is actually consumed in order to change the protein's shape in such a way that it moves things from low concentration upwards to high concentration. So it allows for concentrating a thing. So those are the ones we talk about the most. Those are the channel proteins. We're going to go through them a little bit differently in just another slide here. The top bullet there is how the non-polar things move. They have a much easier time. So if you're a nonpolar molecule, you can slide right through the membrane because you are not affected by this hydrophobic region, right? So you can slide right through. So what does that? Our gases do. So one of the things you will never hear me talk about is an oxygen channel or a carbon dioxide channel, because they don't exist. Those molecules are small, relatively nonpolar. They can slide right through the molecules. Another example of a lipid-soluble or nonpolar molecule are the steroid hormones that are made of cholesterol. Their nature means they don't need a receptor or a channel on the cell surface either. It's one of the reasons why those hormones have such profound effects on our physiology is because they're not receptor mediated. They go right to the nucleus and work there. So two different paths for membrane solubility. My little graph over there in the corner, okay, so the y-axis is how quickly the molecule can move across the membrane, and the x-axis is the concentration of the substance. So in a channel, we have the blue line, right? The greater the concentration difference, the faster the rate of diffusion. The red line shows us a facilitated diffusion situation, where continuing to increase the concentration doesn't increase the rate of diffusion past a certain point. You see that? So that's an example of saturability, like I said about the glucose in the blood. When our blood glucose hits 300, we start to see glucose appearing in the urine. Why? Because the speed at which it can be moved is no longer sufficient to get it all out.